we are starting the lecture. Otherwise, it was previously scheduled by the Professor Jain, but now he will take the 2 hours continuously classes, and this first lecture will be taken by me. The topic of this lecture is the utilization of the NMR for the metabolomics. So you know very well, NMR has a specific word, NMR, N for nuclei, M for magnetic, R for the resonance. So this is a technique that was developed around 1950, and now it is well developed. Previously it was 100 hertz, now it is around 1.2 megahertz. That machine is developed by the different persons. So I will explain the concept of the NMR, but before that we have to start what are the issues with the NMR. As yesterday Professor Jain told that with respect to if you have the parameter, that is the specificity. So NMR has no specificity as compared to the LC-MS. So LC-MS is something that is in better resolution, but with respect to the development of the protocols, development of the different parameters of the NMR, now we are on a stage that we can use the NMR with very improved specificity. So NMR with the improved specificity there are the two things which are installed in the NMR. That's why we can say that we have very good specificity as well as the resolution of the NMR. These two things are associated with the probes actually. So if you go and you will find out that the first issue can be resolved by the cryo probes. So basically what happening, this cryo probes is due to the sample. When you are putting the sample and you are giving a room temperature, so they heat up and they are not able to give the good specificity. So to get the good specificity, we have to maintain the temperature as lowest as possible. So for that, this cryo probe was developed, and this cryo probe is something that can give minus 25 degree temperature due to the supply of the helium gas. So this was a one parameter which influenced the specificity of the NMR. So it was the recent development of the last 5 to 10 years. So now cryo probes are available and you can use it for the different purposes specificity. So another point is that this NMR probes, because sample, how much sample you need the NMR. Previously it was a little bit high, and when you are, it is not possible to use 100 microliter, 200 microliter. So this second development was improving the specificity that is associated with the microprobes. So by using these microprobes, you can use small amount of the sample for this particular purpose also. Otherwise, what happening, previously we used the 1 mm probe, 5 mm probes. Now you can use the 1 mm probes. I will explain everything in a very way, because the starting with the disadvantage of the NMR and the specificity was the one reason, and I am explaining that how these two parameters improve the specificity of the NMR. So this probes now is available, that is around 1 mm probes, and it is around more than 20 to 50 times better resolution as compared to the primitive NMR. So this is a small thing, but now the another issue is associated with the NMR is the overlapping of the different peaks actually. So I will try to explain what is the meaning of these peaks and how you help to understand how you interpret these peaks. But many times what happening, you find out at a particular site, you will find out at a particular site more than one metabolite overlapped with the another metabolite. So this is another issue associated with the NMR. Now we try to find out this thing. Basically NMR has so many variations. We would like to go and read 1H NMR, and I will explain what is the meaning of it also. So in 1H NMR, we have to resolve the overlapping of the different peaks. That is a important point. So these are the different differences between the LC-MS and the NMR spectroscopy. LC-MS is something that is giving the better resolution between the different analytes. And you are using the chromatography, you are using the MS. Two machines are integrated with each other, and they are giving you the best possible coverage of the different metabolites at a particular event. But here, NMR is only one machine. And what you are using small amount of the cell. They are not separated. They are using a different techniques to at which the different environments, electronic environments is giving the resonance. So the utilization, the information, yeah, the techniques that is used, harvest to get the information of the NMR is totally different from the LC-MS. Resolution is a point, but it can be resolved by many other things. There is another important point is the reproducibility. So reproducibility in the NMR is really very good. If you get a clear-cut pattern of the spectra by using the NMR, so if you do the same NMR anyway here, so you will get the same pattern. And if you have only one replicate, that's enough NMR. But for the NMR, you have to use the multiple technical ways to read all the biasness that is caused by the machine. So the different points, measurements is a one sample recount, and LC-MS is a little bit, you can say that distractions, but this spectroscopy have to use the two different. You use the liquid sample also, you can use the solid sample also by the but those are not possible with the LC-MS. In the LC-MS, you have to go ahead, you have to distractive technology, and finally you are not able to use those components for any other purpose. But NMR, if you are using small amount, the rest of the amounts you can use and for the different purposes. Now the targeted and the non-targeted, these two words was covered by the Professor Jain in the yesterday class. So targeted and the non-targeted, these are the just two different types of the class. As I know, the best work in the metabolomics was developed by the Professor Penn laboratory from the USA. In the plant metabolomics, he was the father of the plant metabolomics. So now, targeted are those metabolites which you know that you want to investigate in your work. So you know their structure, you are just looking their pattern at a particular biological situation. So that is possible. We can say that is the targeted. Means those metabolites are already reported by someone. They are submitted in some library, and you are just comparing with respect to that particular metabolite. But in the non-targeted, you don't know anything about the biology of that cell. You don't know anything about it. And you have to start. You don't know what is the meaning of those peaks which are coming here. So that is the non-targeted. Non-targeted has multiple hundreds of the peaks, and you have to analyze. It is a challenging task. But targeted, because if you are just looking some few metabolites, suppose you are working the amino acid, so you are looking the essential amino acid, lysine, knowing the different biological situations. So that is easy. And that both the things you can do by using the NMR spectroscopy. Yes, you can use both the spectroscopy. This LC-MS is also by targeted and the non-targeted. But if you are using the LC-MS and you are using NMR also, and if you can validate both the data, so it shows the validity of your data. How much your finding is the strong. It can be done by any machine, you are getting the same pattern, so it means you are really captured the biology of that particular biological situation. So now I am coming the application of the plant metabolomics in the NMR. So I categorize the whole application of the NMR into the three different categories. The first one is that classification and the characterization of the different plants, in bracket medicinal. So what is the meaning of it actually? You know, we are the different from each other. We have the different facial features. We have the different phenotypical features. But we also behave differently. Our way of talking, way of thinking, thought process is totally different from the another one. Even the same brother, offspring also have a different thought process. So those biological plants, we can classify them on the basis of the biological nomenclature. But every plant has a unique different type of the metabolites. That's why we are using those plants for the different purposes. So every plant is different. And now we have to, maybe in the future after 50 and 100 years, identify that we are giving the nomenclature to the plant based upon the what is the metabolites which is unique in that plants. Maybe you will find out some biological nomenclature, some name of those metabolites. So metabolites is just like a facial feature of that plant in terms of the at the biochemical level actually. So this is the first application, classification and the characterization of the different species of the plant. You can use this word for any organism also. Everyone has its different biological signatures. That's why NMR we can use here also. The second is the monitoring the stress situation, monitoring the developmental clues, monitoring any biological situation. So plants is just like us. Every day we are facing some hurdle, challenging, and it is not possible how we will do it. But plant is not thinking so much. Plant is believing in the response, not our thinking. So how they respond, they just know what is the situation and how they have to adapt those situations. And plants has the better adaptability than human beings also. We don't have the adaptability, that's why we are roaming from one place to another place. So plants, how adapt themselves in those situations, that is their physiology. And physiology means water potential, first thing, and second is the secondary metabolite. So those secondary metabolites which is responsible to help to cope up that particular situation, we can monitoring those things also. If your experiments is come in this situation, so you can use the NMR very well. The third most important is that, okay, we want to make some differentiate, not on the basis of the physical feature, on the basis of the psychological, how we think about the world. Same, if we are developing some cultivars, if there is so many different genotypes, yes, some transgenic, non-transgenic plants, and you want to find out what is the difference, how you differentiate one from the another one. So basically we difference on the basis of a few K metabolites. Those K metabolites we can label as a biomarkers with respect to that particular situation. So biomarkers means something which can be used for the biological variation with respect to the another species, cultivar and the genotypes. So you can screen and identify those biomarkers also by using the NMR. So these are the three different applications we can use using the NMR technology. So the first points, I'm starting from the first point actually of the NMR, and I will cover up to the end of the NMR. So first point that we have to think of your work. Whatever you want to do the work, first mandatory situation is the experiment. You have to design the experiment. Now experiment means the point is that what you want to do actually. So which metabolites you want to identify in a particular situation, or differentiate between the transgenic and the non-transgenic, or you want to differentiate a particular species from the other one on the basis of a particular metabolites accumulation. So yes, whatever we are experiment we design, we have to capture the biological variation in terms of the different biochemical accumulation. So that is our mandate actually. Metabolomics means biological variation. That we want to show that this experiment proved these type of the biological variation as compared to the another samples. This is the one point actually. The another points that come here, another most important, which plant variety you are working. Remember, every plant variety, every cultivar, even the two plants in the same pot, they have the different identity. Every two seeds, they are the different from each other, as genetically and the biochemically also. Genetically, they have some recombinations, frequency differ from each other. That's why two seeds are never similar. Similarly, two plants which are growing in the same pot, they are, according to broadly, they are the same leaf shape, same physiological process. But biochemically, maybe one has some infection, another don't have an infection. Yeah, yet not that infection reach up to that plant. So they differ from each other. Now, the point is that if you are designing some experiments, so you have to fix yourself with a particular variety, particular cultivar, particular genotype, and you have to fix the area from where you have to collect the sample. And remember, those area has some specific shadow. They have, even if you think that 365 days are the same, so might be you are the wrong. Every 365 days are different from the another days. So, hours matters. At what time you are collecting the data? Because there is some metabolites like malic acid. Malic acid is continuously changed within 1 hour. Their pattern, temporal pattern is totally different from the other one. So you have to fix which cultivar, okay, what is the area from where you are collecting, what is the sunlight pattern in that area, and the another point that comes here, harvesting time. So at what time you are picking the sample? Because you are just differentiating your sample from the another one. So you are capturing the biological variation. And these are the different parameters which influence the biological variation. So first of all, you have to optimize this thing that if you are working 2 days a day, so next year if you want to collect the sample, so you have to fix yourself on the same date, same time, early morning. So every sample replicates and the other biological experiment you have to collect from the same time. If you are thinking that nothing will happen, maybe you will get something different and you are not able to find out those biological variations with respect to the human matrix, then your experiment is failed. So you have to emphasize yourself to get the good reproducibility, and if your labs and your work has the good reproducibility, so your research is validated. So reproducibility matters. The another important points, you can use whatever you are collecting today. If you optimize the condition, next year, after 2 years, after 4 years, you already develop a reference with respect to those conditions, experimental conditions. So that you have, if whatever you are doing, maybe the same thing can be used for as a reference material in the near future. So whatever the experiment you have to do, please take each and every one very sincerely with respect to the metabolomics. Okay. The another points, you have to store very well metadata, because the next year if you are doing the same thing, so 2-day data, stored data can be used as a reference material for the next one. Now the replicates. How many replicates you need for the NMR and how you have to go ahead with this one. So basically there are the two things. First is the number of the plant per sample. Number of the plants per sample means in one sample how many plants you have to take. What we are doing, we are using one plant, one sample. Okay, every plant is different and there will be minor differences. So every sample, whatever you are taking, it must be more than one plant sample. The source must be from more than one plant. So that there is a little bit difference between the two plants, it can be minimized. And you only work with that biological sample. So what we have to do, we have to collect the sample from 5 to 6 plants, mix them so that we can minimize that type of the errors. So we have to do these things. Second is the biological replicates. Biological replicates means how many samples you are collecting for a particular experiment. So biological sample means today you are collecting 5 samples, so you have to separate them into the different sets, and every set have the same type of the sample. Those we are tagging them as a biological sample. So basically, if we are using the different points, actually different time points after the infections, if we are taking the infection, so it varies. I would like to recommend it, you have to go ahead 4 to 7 replicates biological to track the biological variation of your sample as compared to the another sample. That thing you can do here. The another point is the technical replicates. If you are working with the NMR, so no need to take the technical replicate, because NMR one spectra is enough to give you the clear picture of your work. And this biological replicates is more, so you have to emphasize yourself for more biological replicates than the technical replicates. Someone asked me, where is the statics in the work? Technical replicates, one sample is enough for the NMR, but if you are working with the LCMS and the GCMS, so So I would like to recommend it, at least 2 and the 3 technical applications you have to cover for your work. So here you know that how many samples you have to put in, how many plants you have to put in one sample, how many biological replicates you have to do, and how many technical activities. Harvesting time, as I told you. So you have to cover fresh materials, you have to take care of. What is the meaning of the fresh materials? Yes, when you are going, so you are just taking the leaves, okay, roots, stems, and you are putting in the bag, and after 24 hours you want to do the metabolomics. So sorry to say, your sample is already destroyed. These metabolites of any cell is changing every second actually. When you cut the leaf, so leaf started to respond because plant believe in the response. So when any situation change, plant change their response, and response means the whole biochemical concept of that plant is changed. So you are capturing something different, and you have to capture that particular, you are capturing a particular reaction, so you have to first of all freeze the sample as early as when you take from any plant. So many times what we are doing, we are taking the whole plants and putting somewhere here, and just, we are not, first of all you have to find out which tissue you have to use for your experiments. Is it root, stem, or leaf? The second point, if you are taking the leaf, so I would like to recommend you, please avoid the veins, because veins has different type of the biochemistry than those mesophyll and the palisade cells. And yes, if you are looking, you have very small amount of the sample, only very small amount of the sample, just only 5 microgram, 10 microgram. So you have to focus that whatever the cell is present in that sample, they are highly homogeneous. Sometimes, you know, cell present on the skin, cell present hair follicle cells, they are the totally different from each other. So if you are putting the skin cell, so sometimes hair follicle cell is also come in the skin cell. So first of all, you have to remove the hair follicle cell, then use the skin cell for any particular event. These skin cells are dead, but the cells, living cells just below the skin cell, they can give the better availability of the biochemical which are also nourish this thing. Our cells which are present just below the skin, they can get some exchange of the gases from the dead skin also. So you have to try to understand if you are working on the embryo cells, so you have to remove all the cotyledon parts and only just focus the embryo for the extraction purposes. So homogeneity of the sample is more important if you have very less sample. Because more homogeneous cell means you are able to capture those biological variations with very accuracy. So you have to take care of these things. The another point is that whatever the sample you stored today, you have to use within 10 to 15 days. If you think that next year we will use, so your biological information is already lost. So if you today collect the sample, and remember you have to collect the sample in the liquid nitrogen. Liquid nitrogen freeze the sample at the same time. Now you are able to grind your sample. So those things are happening here. So sample preparation, what the samples you have to take, and how to use for your work. So there are the 3 things. First is the drying, second is the weighing, and third is the extraction. So drying and freezing, this is a concept. Drying may you have, what is the meaning of the drying here? Every cell has 70 to 80% water molecules. And these water molecules which are present in the cell, they are the good source for the enzyme to do the different enzyme activity. Okay. So if you are taking the sample and that is not yet not dried actually, so what will happen? Those enzymes are highly active and they catalyze the different reactions, and you are getting different type of the metabolites. Every enzyme is, some metabolites are the key substrates for the enzyme, and they can produce the different products also. So what we have to do? We have to collect the sample in the liquid nitrogen, and we have to dry them as early as. Second sub-stage of the drying is the grounding. You have to grind the sample. So we do both the things in the one step. liquid nitrogen freeze thawing methods, in which what we are doing, we are grinded the sample at minus degree temperature, minus liquid nitrogen. So when you grind the sample, and yes, there is one thing that happened, you can remove the water by at the low pressure. So what we grind the sample in the liquid nitrogen and we put the sample at the low pressure. So what you liquolizer, in the liquolizer, so whatever the, with the liquid nitrogen, whatever the water molecules are present in those samples, we try to dry the sample by removing those water molecules. But remember one thing always, we are not able to remove all the water molecules from any sample. Water molecules are also present with many hydroxy group of many carbohydrates and amino groups of the proteins. We are not able to remove those things. But yes, they are not able to active the enzyme. So we have to minimize the water molecules in the sample, and we are not able to completely remove the water molecules from the sample. So this freeze drying method is the best one. Now you know dry sample, powdered sample. The second point is the weighing. Weighing is the most accurate way to get the accuracy in the biological capturing of the values. If you are weighing 50 mg, 100 mg, so you have to go ahead with 10 to 20 mg weighing, accurate weighing of your sample, infected samples of the experiment, and the control sample of your experiment, and you have to use the equal weight of the both the things. And it would be better if you go ahead with the replicate scale. Little bit difference in the weight is very difficult to capture at the later stage. Otherwise, you have to use robust chemometrics tool to normalize those cases. The other extraction, so extraction you have to do. They are the two different things that come up here. So this is the same thing that I told you about the water. And freeze drying is the method that we are using for this particular purpose. And material sublime under the low pressure, so whatever the water molecule is present, it can be washed. So this metabolites could eventually degrade. Yes, if someone say that, if this is the best method, yes, this is the best method, but many metabolites are also degraded during this process. But they are not influencing too much our work, so we can ignore this thing also. We can't use the significant loss. This is a minor loss, and we can ignore this particular things. Now coming on the another point, that is the weighting and the extraction that I told you. Extraction means you have to select the solvent. I am just only discussing about the NMR, remember. So extraction means, yes, every metabolites differ from the others on the basis of their weight, on the basis of their ionic strength, on the basis of their structure, and so much diversity with substitutes. So they have some are the hydrophilic, some are the hydrophobic, so some require the organic solvents, some require those normal solvents. So if you are looking what type of the metabolites you need to extract, for that you have to use only a specific type of the solvents. There is so many types of the solvents, and you have to find out which one is giving the best to you for your work. So perchloric acid is basically, extraction is the most suitable for the polar components, because polar means which has hydroxynuclea, which is hydrophilic, you can say, like the sugars and the amino acids, because they have the substituents, but it is not suitable for non-polar components. And the another point is that if some compounds is acid labile, so we have to avoid the using of the perchloric extraction method. The second is that aqueous methanol is used for the polar components also. I'll just explain too much because I thought that Professor Zeng will tell about the extraction protocol, so we skipped that thing. So I just give you the overview of this thing. Aqueous methanol, you can use the single extraction also, and you can use the double extraction also. In the single extraction, methanol is basically the most important components we are using in the lab. Remember, you don't go ahead. If you are working highly specific, then you have to select the actual solvents. Otherwise, go ahead with the methanol directly. And remember, you are using, and in how much dissolve in how much methanol. This thing matters. Please take care of this. So aqueous methanol you can use with the combination with the chloroform also. Yes, if you are using with the chloroform, so you are covering many components which are polar as well as the non-polar. So you will get the better coverage of the metabolites if you are working in the two solvent system, in which one is the used for the polar, second is used for the non-polar components. Now pH measurement, pH of the extractions. What is the pH of the extraction actually? Basically, if you are controlling the pH of the extraction, so you can avoid the shifting in the NMR. I will explain what is the meaning of the shifting in the NMR signals. So many buffers we have to use for the NMR, and phosphate buffer is the most useful to maintain the pH of your extraction. So if little bit difference in the pH now, so your whole NMR spectra is shifted and it will create the problem for you for the analysis of the data. Yes, sometimes there is an issue. We have to avoid the methanol, we have to avoid the chloroform. We are using NMR solvent. So I will explain what is the meaning of the NMR solvent, then I will help to know what is the NMR. So this is the 1H NMR, and this is the, you can see here, there are the NMR spectra. It is of a plant, and you can see here, it has the three different regions. One is showing the amino acid region, second is the sugar region, and third one is the aromatic region. And remember, you easily get the NMR within the 30 minutes. So one day for the sample preparation, and next day you can go ahead with the 1H NMR. You get the spectra, and these spectra are just like this one, and this is the case of a particular Arabidopsis plant which has sugar, amino acid region, and the aromatic region in a particular extraction protocol. Yes, this is another NMR. The absolute concentration of the metabolites can be easily calculated by using the NMR. So this is the same thing that I told you, but you know here there is a 1 point come up here. I told you this is the 1X. So basically this is the internal standards that we have to put in the sample before to run the NMR. This internal standards, I will explain what is the use of this one, but we are using this chemical shifting, the number numerical, here it is a 0 actually. So basically you can't see this IS. Whenever you find the 0 ppm, so that is the internal standards. Okay, whatever sample you will find out, it will be between the 2 and 8 ppm. But I will also explain you what is the meaning of the ppm, delta, and the chemical shifting also. So yes, but sometimes we are using this internal standards. We have, this is a sample that is NMR internal standards. So we have to identify the different peaks with respect to this internal standards only. How much they are different from this thing. Okay, and yes, there are another points. Sometimes if we are using macromolecules, very big carbohydrates or something, so they broaden the peaks actually too much. So then, so what happening actually, when they broaden the peaks, so peaks, upper side is okay. Actual peaks is just like this, a single lane basically. But when they have some broadening and some another issues, so they will come up with this. So basically in the carbohydrates and amino acids, you can see the broadening. Now these broadening, we can reduce those things by just manipulating the parameters of the NMR. I thought that it was here, but might be that is not here, so I will give the parameters for the different spectroscopy in the next lecture. So basically to reduce the broadening, we are using these two different parameters actually. These are already optimized in the different NMR, you have to use one. Manual modification in the car versus spin eco-sequence methods, which can be used to increase the resolution basically. So those will come up. Yeah. So now the another points come up that, as I told you previously, we are not able to. This one. No, okay. these two methods, whatever we are using, they can help us to get the coverage from the low molecular metabolic cells. That's why we have to do this thing, NMR pulse sequences. We have to take care of these things. Pulse sequences means parameters. What there are 7 to 8 different parameters of the NMR which we have to optimize according to the different conditions and they give the better resolution. On the basis of that, you will get the different coverage. The another point that come up that whatever the water molecules which is left in the sample, it was difficult to, as I told you now, because they are associated with the carbohydrates, they are associated binding with the amino sugars. So yes, water molecules are present even in your sample when you extract it with the anisole. So we have to reduce those signal which is caused by those water molecules by using the suppression methods. So suppression method by which we can make those peaks as a 0 one. So whatever the new pattern come up in which we are not able to find out the signal caused by the water molecule. So that is easy for us for the analysis purpose. So these are the popular extract water suppression methods which mentioned here. The first one is that water rate. These are the methods actually. You just modify the condition of the NMR. You can easily remove those things automatically by that NMR machine. So water gauge is one of them in which they are doing some manipulation of the different parameters, and that is the water suppression by gradient tailored excitation. So this is a resonance, so they are excited the resonance in a gradient manner. Combination of the gradient and the weak radio frequency pulses, that is the wet water suppression enhanced through the TI effect. This is another methods by which we can reduce the water suppression. And the third is the preset. Preset is the most adopted method actually. So you are already used. We are already used it many times and we are using it due to its simplicity. आप उसको पहले से सेट करके इधर निकाल देते हैं. Then you can use it. So basically, when we use the preset, so you don't need to bother about the water peaks. There is already suppressed. Now you are getting the peaks of only your metabolites. So now before I have to go ahead with the NMR data, what type of the spectra you are getting. So I would like to take 10 to 20 minutes about the NMR. Until you don't know about the NMR, you are not able to understand the spectra of the NMR. So NMR is just like that I told you, NMR, nuclei, magnetic and the resonance. So your nuclei is present in a magnetic field and before magnetic field, and we are just manipulating the alignment of that nuclei. So when you manipulate the alignment of that nuclei by using some resonance, so they release the observations and that observation we capture in the form of the observation spectra. That is the concept of the NMR basically. So this is a question that I would like to ask myself, not you, how this spectroscopy is useful about the structure of the organic molecules. So I am taking here as organic molecules as an example and try to understand how it gives the structure of that particular organic component. So basically, chemically, non-equivalents, protons, have different resonance frequency in the same applied field. So in a layman, I can say that our friends and me behave differently in the same situation. Okay, there is a situation in front of me. I will behave differently. We will behave differently. Okay, but if my sibling come with me and we are nourished in the same way, so maybe we both behave in the same way. Okay, so that is the major thing I would like to say here. So suppose this is a component, that is the methyl acetate. It has because this is the 1H NMR. So here we have to consider only the proton behavior. Okay, and proton behavior means here this is the one methyl acetate. So it has two different houses in which these protons are present. And these two houses are totally different from each other. One is this one. This is a different environment. This is the another environment. So we can label them by the two different non-equivalent protons that, because many of you are from the biological background, so maybe you don't know about the chemistry. You come after graduation so many years. So I'm using the name and language for the better understanding of this concept. So these two parts of this method, two parts of these compounds have the two different environments. One environment has a different type of the proton, second environment has a different type of the proton. So this NMR will give us two peaks of these two different environments. So basically what happening, whatever the nuclei, these two nuclei of the two different neighbors, when we put in the magnetic field, so suppose this is the magnetic field applied, and this is the particular nuclei, and these are the three similar nuclei. So what happening actually, so at first the magnetic moments, half of the proton, whatever the half proton, aligned in the opposite, aligned in the pair, in the same direction. So half in aligned with the magnetic field, half aligned with the opposite field actually. So what happening, the sample is, when we expose them, when your sample is fixed, aligned along with the magnetic field, half magnetic field, half opposite to the magnetic field, and if you expose your sample, now the electromagnetic waves, so what happening, your half non-aligned sample also aligned, and after some time they try to go back in their natural alignment. So this disturbance that we create by supply of the electromagnetic wave, that is labeled as a resonance actually. So we try to capture this resonance by using this method, and this proton absorb radiation of this, they are giving at the two different resonance frequencies. So we are getting the two different radiations at the two different resonances. So NMR instruments record those different frequencies which absorb as well as the intensity of the each resonance. So NMR is just doing those things. Whatever the molecules behave differently when you apply the magnetic field with supplying the electromagnetic wave, we try to capture those differences by using the NMR. Now, most of the sample, you know, we are taking the solutions. So most of the NMR solutions, but you can do intact plant also by using the HR mass spectroscopy, and that is also in the research very well used by so many researchers. So mostly solvents, because we are here about the solvents, so as I told you, which solvent we have to use. So solvents means we try to use those solvents in which H is replaced by the deuterium, basically, because we want to avoid all the peaks of the bottom molecules. So yes, we can use whatever the problem, that's why. So we use the specific type of the solvents for this particular purpose. The first one is the deuterated fluorophore, second is the B6 acetone, and B2O. Here there is no H. Because now, what is the importance of this component is that deuterium is NMR active actually, and it has a resonance that is very difficult from that of the proton. So what it is invisible in the 1H NMR, and it invisible means, so there is no influence of the solvents we are using for our sample, because whatever we sample use, we avoid whatever the by other influence of those sample by some solvents. So we have to use such type of the solvent to avoid any misinterpretations. Basically this is the things come up. So 1H NMR float for the methyl acetates has the two different components. This is another environment, and this is the another requirement. So you are getting the two non-equivalent protons, and they are giving the two different peaks. One peak is here, and second peak is the here, and they are almost similar height. So what is the meaning of it? It means both have this non-equivalent proton population has the same number of the proton. 3 here, 3 here. Now, and this is the internal standard. So these two peaks are associated with the one is with the HA and second is with the HB. The third right peak is internal standards that I told you. Basically, it's very difficult. when you get the NMR spectra, you will never get the TMS. It is always starting with a 0 1. So 0 means that peak is associated with the internal control. So this is another component. It gives the TMS. TMS is an internal standards here used, and it has this particular structure. So why we have to use the TMS? Now I am coming on the points actually. That is the basics of the NMR for all of you. And what is the meaning of this ppm? Whatever is here written. What is the meaning of the delta? What is the meaning of the ppm? And why we are using the TMS? These are the associated things along with the interpretation of the chemical safety. So look here, why we need the TMS and what is the meaning of the delta? So let me give you an example. If you want to use the parameter that is the hertz. So resonance, electromagnetic wave, you know very well, hertz. So previous NMR of the 100 hertz, 1956 and 1950 NMR come up in the first time. So 100 hertz. Now you see it's 600 hertz NMR we have here, 600. But right now it is around 1.2 kilohertz. NMR is present in the many laboratories. So hertz, if you are using the hertz as a parameter for the resonance, so we have 100 hertz machine, okay, and Dr. Atis has 600 hertz machine. So those machines are giving the different type of the patterns, because my ability to excite the electron only about the 100 hertz. This machine has the ability to excite the electron, the nuclei is much more powerful than me, manifold powerful. So when you collect the data, so it's very difficult to equilibrate them. There is no interconversion concept which can convert his work, his pattern along with me. So you are getting the different, every different machine is giving the different chemical spectra of the same experiment, of the same data. And you can't convert one data into the another one. So hertz is not a good concept, basically. Now, this is the same thing that I told you. You can use the 300 megahertz instruments, and someone is using the 100 megahertz instruments, and both of them are giving the different type of the concept. This is a Tesla magnetic field, capacity of the magnetic field 7.1 Tesla, this is the 2.4 Tesla, and they can resonate any protein. This is the resonance power up to 100 megahertz. So this is the capacity of that particular NMR. Another NMR has more good one. It has 300. It can resonate the molecules up to the 300 megahertz. So we have here NMR that has the 600 megahertz. But right now 1200 megahertz NMR is also present in many laboratories. So these, now you get the different type of the data from the one. You are getting the different type of the data from the another one. So now it seems to be very difficult for all of us. How could we go ahead? So we avoid NMR and inconvenient and confusing situation for all of us to convert the NMR data from one machine that has 100 megahertz and second one has the machine that has the 300 megahertz. So that's why we can't compare the data on the basis of the hertz at least. So now what is the another point? If you use an internal standards, in here machine which has the 100 hertz, megahertz, second machine has the 300 megahertz. So you will find out the internal control is come up with the 0, and whatever the pattern you are getting, the difference between the internal control and those peaks are the same here in my machine and the same Dr. Atis machine. So we avoid the megahertz concept for the NMR spectra, and we have to fix ourselves with the ppm. ppm is the parts per million. So what happening? These are the 12 protons which are giving at a particular situation peaks, and we are tagged that peak area as a 0. So whatever the metabolites come, we have to calculate their distance from the 0 actually. It will be the same. If the machine is present in the North Carolina, machine is present in the Maharashtra, and they have a different potential, different Tesla and the different megahertz, so they will give the same pattern if we use the TMS as an internal standards and we calculate the thing with the help of this particular parameter. This is the same thing that I told you. The resonance frequency of the proton is the same, sample molecule is the same. How much higher they are, so we are using the ppm. So ppm means 12 molecules of the TMS is down here, so they are giving the 0, and after that whatever we are getting, we are tagging them. So this is the points here 0 and these are the 2. So these two proton groups are giving at the different situation actually. Now you know what is the meaning of it. It is the 2 ppm, 2.05. It is the 3.67 ppm. And now you know the meaning of the machine also. What is the meaning of the 300 megahertz? And so 1 ppm, remember 1 ppm is equal to, of the 300 megahertz is equal to the 300 hertz actually, 0.1 ppm. So 1 ppm, if you are going this way, so molecules resonate at the higher frequency. Okay, so that's why this is the least one, that is the 0, and whatever the molecules moving this way, so their resonance at a higher frequency. So that's why this ppm is the number of the protons which are giving the response at a particular site. Okay, now I'm going back a little bit so that you know about the carbohydrates and protein. So it was the thing. So you know amino acid regions, it is near to the 0 ppm. Then sugar are more complex actually. They have more, they show the resonance at a very hard situation. And then aromatic region, aromatic means secondary metabolites and many other things. They require more energy for the resonance. So that's why more powerful resonance means they are far away from the 0 actually. So this parameter, what a ppm, is the most important for all of us. And whatever the pattern you will get, it will be in between the 2 and the 8 ppm. So 2 and the 8 ppm window is the area where you will capture the maximum resonance in a particular NMR. This is the point that I would like to explain here. And I know a little bit you know now about the NMR. Okay. Yes. So what happening actually, whatever the sample you will take, you have to add little bit amount of that particular TMS, and then what will happen, and you have to put that TMS peak as a 0 points. Now, this practice is generally no longer necessary for everyone. In the modern NMR, they use the deuterium signal from the solvent automatically as a standard solvents. So, TMS now you don't need actually. Deuterium solvent is already come at the 0 position reference points, and you have to use that reference point as a 0 baseline that is corresponding to the TMS proton signal. And yes, this is the same thing. It resonates at a particular CDCl3 is resonate, not resonate at the 300 megahertz, it is resonate at the 32 megahertz, but we are putting it as a TMS signal. So in the remaining NMR spectra, whatever remaining part of the NMR spectra, we will not see any actual TMS signal. Whenever you are getting the NMR spectrum, you never saw the TMS signal. We can always assume that 0 ppm is just like corresponding where the TMS is giving their actual proton resonance. So now TMS we are skipping. So computer in the NMR instruments can be instructed automatically to integrate the areas under a signal and the group of the signal. So here as the same thing comes again, you can understand that these two different protons are doing the two different things, and it is associated with, so one HNMR is basically a signal that is proportional to the number of the hydrogen which has the peak corresponding to. So one HNMR is only associated with this hydrogen proton resonance, and two signals in the methyl acetate pattern integrate to approximate in the same area because they have the same number of the equivalent protons. So you know little bit what is the NMR spectra, what is the meaning of the peak here. Now I am coming little bit more. So paragyning is another concept. It also gives the two peaks, but one peak is higher than the another one. So this peak is higher as compared to this one because they have the non-equivalent protons of the different number. One has only 1.5 higher because one has the two set of the protons, 6 methyl protons and the 4 aromatic protons. So 6 methyl protons and the 4 aromatic protons are coming at the different points and they have the different heights also. But in the previous case, heights of the two different proton number were the same. Here they are not same because one has the 4, second has the 6 second. So that's why they up and down. So integration function can be used actually to determine the relative amount of two or more compounds also. So if they are the 4 protons, so peak is up to here, they are the 6 protons, so peak is up to here. And the second, if the two different chemical species are present, if you fix them, so you can find out the exact concentration in mixture of both the different constituents also. That thing can also possible by using the NMR. So here 50-50 moles, here 50-50 moles of the acetone and the cyclopentane. If you are using those mixtures, so you can easily identify what is the exact composition of those mixtures by using the NMR. Remember, if you can find out the composition of the mixture, then it is easy for you to find out the concentration of the different metabolites which are present in your plant extraction complex. Now it is easy for you. So now this is the spectra and NMR spectra, and yes, it has the 3 different parameters that we have to take care. The first one is the integral, multiplicity and the chemical shifting. I will little bit explain the chemical shifting and avoid this integral and the multiplicity because we have to cover many parts and we have how we analyze the data, that is the most important for us. So in this chemical shifting provide about the different environments in each proton. So what are the protons which are present in that particular sample and how they have a different chemical environments. So every chemical environment will give one peak actually. So here you can find out how many protons and how many different chemical environment of those protons by using this NMR spectrum. Yes, simplicity, this integral is another component. Multiplicity is a reflection on the number of the non-equivalent protons located on the adapted carbons also. So those things you can also calculate. And third one parameter, that is another point, that is the integral. Area is the relative number of the proton signal. One proton gives signal here, 2 proton give the signal here, 3 proton give the signal here. So that type of the parameter we use the integral. So chemical integral and the multiplicity, these are the 3 types of the information you can retrieve by using the NMR spectrum. Now these are the different functional groups which can give you the different type of the area in the NMR spectra from where you can cover. So you will find out more complex components has spectra far away from the ppm. Okay, this is the more complex as compared to all those. So they are giving the ppm at the 12. And as I told you, you have to cover 2 to 8 maximum up to there. You are covering here proton behavior. Okay, what about the compounds? But the different substituent with the protons are giving the different chemical behavior. So that's why it is possible for you on the basis of the non-equivalent, on the basis of the behavior of the proton in the different chemical environments, you can find out the different type of the metabolites. So these are the different functional groups which are giving the different environment to the protons, and that's why we are able to identify those components. So now, in plants basically, there is a standards which is developed by many laboratories. So one of the laboratory which maintain this one is the 2 different metabolites. So 20 different metabolites are already, these are the reference material. You can use, anyone can use this one also. So this is the identification of the metabolites in the metabolomics analysis is the most important and the same time the most time consuming steps. Now what happening? The first step is that identification of the metabolites in the assignments of the resonance. So comparison is the comparison of the NMR chemical shifting also and the coupling constraints with those of the authentic samples also. If you know some standards, if you know some authentic samples, then it is easy for you. So many times now NMR can be used to find out the adulterated in some components also. Many one is selling many herbal medicines, many other things. One of the things I know that is a mineral, rock mineral, that is the silagi. You will find out 95% adulterated. Second is the saffron, and 95% is the chemical synthesized component. How you differentiate them? If you know the authentic saffron, you will get the different kinds of mix, and you just do the NMR spectra. You can say that this sample is authentic or non-authentic. So for that you have to first of all have the NMR spectra of the authentic sample. So NMR is further, we are classifying the NMR in the further two categories. First is the one-dimensional NMR, second is the two-dimensional NMR. One-dimensional NMR and the two-dimensional NMR of the, there is so many available databases are present on both the NMRs. Okay, so you have to find out you are working on which metabolites, which laboratory is already generating the databases on those metabolites. Those are useful for you, remember. And these are the different metabolites which have the different signal actually. So these are the data, these are the conditions, and you will find out the J of each and every metabolite. These are the most common metabolites which can be NMR, which can be recorded very well by the NMR. So this is something which can be used here. Now, identification of the unknown. If you compare and you get a known compounds, working with the known compounds, then it is easy for you. But if you are working on the unknown components, so that is a little bit difficult for you, and that requires so much time. So for that, we have to go with the two-dimensional NMR. Two-dimensional NMR, we have to use the another thing. The plot is between the 1H, 1J result spectra. That is very useful for the two-dimensional NMR. Here we are plotting the plots between the chemical shifting versus the coupling constants. So a new thing come up, that is the coupling constants here. So if you go back, this is the chemical shifting, and this is the coupling constants. So one dimensional we just only focus on the chemical shifting, but in the second dimension we are going with the plot between the chemical shifting and the J constants, and this is very useful for the identification of the signal in the crowded region. The signal of the chemical shifting are projected, and the simplified skyline projection spectra can be generated due to these projections. Multiple signals appear as the singlet. So this is the coupling constants, and this is the ppm chemical shifting. This is the NMR spectra. But when you do the 2D, two-dimensional, so you will get that is the singlets. Every singlet has multiple signals also. And this is something which can help us to identify the unknown samples many times. So this projection sample can be further used for the multivariate analysis. So I have not too much time, but I have the time for 2 to 4, in which I will little bit explain about the multivariate analysis also and many other things also. So this is the two-dimension. I just give you the overview. Rest of the things I will cover in my next lecture. So these are the different. Whatever the data you collect, so these are the pre-filter you have to use for your work. And after that, we have to use the multivariate analysis. So this is lecture 30 minutes. So I will cover in the next lecture. You have to do this thing. And after that, this is the final data you will get. On the basis of that, you can identify your sample is totally different from the other one. And you have to identify the sample. And you have to do this thing. So, thank you for your patience and listening carefully this informative lecture. Yes, this lecture we just prefixed. Now, I would like to invite the Professor Jain for the next lecture. And
so, 1 ppm is equal to, you must be 300 megahertz or 300 hertz. So, megahertz is 10 to the power 6, I think.
we will discuss everything in the tutorial. उसमें हम सारा discuss करेंगे. Yes sir, please. Professor Jain. Yeah. It's your turn now. Okay. So, sir will explain the two things. One is That is the LCMS, and second is the multivariate analysis. No, these things differ from every case. The first thing is that what metabolite we have to analyze. Basically, this extraction protocol is very tedious. If you are using another paper who is already published, so it's okay, you will get the same pattern. But if you optimize your extraction protocol yourself for your own work, that is ammonia, some metabolites, so you have to use array of the solvents in the different dilutions also, different solvent and this organic solvent and the polar combination also, two component system, sometimes we have to use a three component system also. You will identify only a few metabolites and those can mix during these two steps also. So yes, if I get a chance, so in tutorial I will explain how a small shift in the pH, influence the peak actually. So I will come with those slides also. I will cover first of all my NMR portion, that is LCMS and LC. So you know a little bit change in the pH influence the one peak was missing at the 4.5. Okay, when you shift 4.5 to 4.1 pH, the peak come up, and what was the peak that was present previously? It was absent in this case. So a little bit change of the pH can influence so much spectra. So try to understand how much a whole solvent influence. Solvent influence the pH, solvent influence the ionic strength, and solvent influence the coverage actually. So first part is the coverage. What coverage of the metabolites you consider for your work? If you want the maximum coverage, then you have to use first of all the hydrophilic polar, extract the polar, then you have to cover the independent non-solvent, non-polar solvents also, then you have to use the combination also. So you will get the different patterns, but we don't know in which pattern you are, what peak you are looking actually. So solvent extraction itself is an art, and it requires robust patience, because otherwise how could you know that what peak is what? So that's why whoever is developing method of extraction protocols, they deserve the first authorship equally in the papers. And if you are giving some sample to the private company, okay, metabolomics services, they will give you the one condition. If you give them, you give the method, they will run. They never need any authorship. But if you say that you develop your method yourself, okay, we are giving the sample only, then they will also ask for the authorship. And remember, in those machines, the most important experiment is the method development. Method development is itself you will get a data. And if you get a very good method, then the rest of the things is easy for you also. So, because we just covered the different parts actually. So I will try to give you the screenshot of the paper that was published in the Nature protocol actually. They use the different parameters for the different spectroscopy. They give the fixed value. You use this one, so you will get that type of the spectroscopy. If you are using these conditions, so you will get the different type of the spectroscopy. So, and solvents, as I know, more than 120 different combination of the solvents are using in the good laboratory to get the better coverage actually. But because methanol is easily available and it gives the extraction, it gives around 60 to 65% of the total metabolites polar compounds. Solvent does not mean you will get the 100% polar compounds. In the polar compounds, size matters, many things other matter. Some polar compound associated with the non-polar compounds also, like glycolipids and many other things which have very long size and many things. So first thing is that what we have to analyze. If you are analyzing only just a few compounds, then this 50%, 60%, 70% is okay, no issue. But if you are working in others, sir, then it is really a serious issue. So 50, 60, even 59, 60, these two also give the different type of the values. I will explain those things in the tutorial in the class. I will take an example of the LCMS in which you find out a single little bit change in the buffer can induce the whole change in the pattern. Then you understand the sensitivity of those things actually. Thank you. If you know the peaks coming at what ppm, you can analyze what type of the function group is present and how those function groups are pushing the things far away from the zero value. So here this zero value is the TTM, but now if more complex compound is giving the ppm in this direction. So in last presentation you know very well, these are the amino, then there is a sugar, and then more complex compound comes later on. So these are the three components we have to take care always when we have to analyze the data of the NMR. I will explain the GC data analysis also. So here there are the three things. First is the integral, second is the shift, and third is the multiplicity. These three are giving the overall information about the different analytes, the number of the protons, type of the protons means different environments in which they are resonate, and the third is that how much they are apart from each other with respect to the standard reference material. So now these function groups, so as I told you, the different metabolites require the different type of the components. On the basis of that, we have to go ahead with the NMR, that is the proton NMR, and here we have to draw the plot. In one more step we are moving ahead now. We have to draw a plot actually that is the two-dimensional with respect to the chemical shift versus the J coupling. So this when you plot the graph between the chemical shift and the J coupling, so you will get such type of the plots. These plots is little bit different from the previous one in which you are only identifying a few peaks. Here you can find out the singlets, and those singlets and representing the different type of the analytes within the sample also. So it can be used further for the multivariate analysis. Now, yes, there are so many different type of the two-dimensional NMR spectroscopy. NMR is a spectroscopy itself, and whatever the data we are getting captured in the NMR, that is equivalent to the IR spectroscopy. So IR and the NMR have a similar type of the pattern here. Now if you saw that I am explaining here the four different type of the two-dimensional NMR also. The first one is related to the correlation spectroscopy. Here we have to correlate the different type of the variables. So it shows the correlation of the protons which has mutual spin-spin coupling actually. So spin-spin, which all those data sets which have the mutual coupling with each other. So for that we are using the first one, that is the correlation spectroscopy. Second is the total correlation spectroscopy. The first one was labeled as a TOC actually. Second was labeled as a TOC. TOC is very useful for the carbohydrates and the amino groups. Identify typically one typical signal actually as a assigned one, and yes, just like the anomeric hydroxy group that is present in the sugar. The remaining signal even we can identify in the crowded area also, and we can use them for the correlation analysis. The third one, that is the heteronuclear multiple bond correlation spectroscopy. Here you can find out the long range correlation between the carbon and the proton can be analyzed, which can identify the resonance within the same molecule actually. So these are the four different types of the NMR, which we manipulate the parameters and we can go ahead with those NMR actually. The fourth one is the heteronuclear single quantum coherence spectroscopy. It is very useful basically in the metabolomics field, and the presence of the certain metabolites can be easily distinguished from the other one by the spectra based upon some reference compound. So if you know some reference compounds, so we would like to go ahead with the heteronuclear single quantum coherence spectroscopy. This is the variation of the NMR basically. Now I am coming in the two-dimensional actually. If you think that you are accurately identifying the compound in a mixture, so this is not too much realistic. you have to face a lot of the challenges, and still you have to do a lot of the work here to identify the exact precise metabolites which is present in that particular spectrum. So for that sometimes we are using the integrated approach in which we are using the chromatography along with the NMR and HPLC you can say, but when you are using the HPLC with the NMR, so major issues come again that is related to the specificity. So you have to compromise the specificity of the NMR if you are integrating the L03 sample of the HPLC for the. Sometimes HPLC you can use, and HPLC can give you the preparative and analytical. In preparative, what you are getting the elute in which your metabolites is only present. If you use such type of the things for and integrated with the NMR, so yes, resolution is good, but specificity definitely down much more. The another important point is that you can go ahead with another HPLC, SPE, NMR in which you are using SPE, and that is the good thing, and that can be recommended for the further use if you want to integrate the chromatography and the spectroscopy in along with the data analysis. So sometimes NMR is not only single approach which we can use, sometimes we recommended the integrated approach in which both the chromatography and the NMR are recommended for the metabolomics purpose. Now, question come up that how you analyze the signal actually. So signals basically come in the form of the many steps, and this can be said that you have to analyze the data. Okay, analyze the data means you have populations of the data. Some are the large, some are the big, some are the thick, some are the dark, some are the light. So this is the variables. Okay, on the basis of the height, on the basis of the skin color, these are the patterns you are getting. Now question come up that you can only analyze if you have the curated data which you can say that this is the dark, this is the light, this is the height, you measured, then only you can say some are the large and some are the small, and some skin color are fair, some are the dark, because you observe that. So for that you have to, whatever the pattern you have received in the NMR, you have to do the pre-processing before the data analysis. Data analysis you can only do when you pre-process these steps. So these are the recommended steps for every NMR spectra. Whatever the sample you are using, you have to go ahead with these 5 to 6 different steps. The first one is the alignment, second is associated with the base corrections, the third is the bucketing, that is the most important components here, then normalization of the data, and first one is the scaling. So I will explain each and every one for all of you so that you know that what is the meaning of the alignment, upscaling, bucketing, normalization and the base correction. So this is the first one that I would like to address here, that is the spectrum alignment actually. So whatever the NMR spectrum you are getting here, you have, there is some NMR resonance is shifted due to some region actually. So you know at a particular wavelength there is an NMR resonance, but sometimes it is shifted from that particular wavelength to the another little bit far away or near to spectra. So for that, because sometimes temperature matters, pH matters, salt concentration and homogeneous magnetic field also. These are the 4 to 5 different variables which influence the shifting of the spectra. Now you have to manage, and so first of all you have to sort out this issue. Otherwise here you are getting something different, and when you are doing the same repeat again you are getting something different because a little bit pH is giving you the different result. So first of all, spectral alignments, it can be do and it rise the variation among the different spectra. So if you are doing some experiment today and after 2 days you are getting another experiment, so this way you collect all the spectra, so you will find out there is a little bit difference between the different spectra. So that is due to these 4 or 5 regions, and that's why we have to produce some alignment of these spectra. So this problem can be resolved by using the standard chemical drift shift, and that can metabolites and the spectra can be aligned. So when we are using the chemical shifting, so we have to take the chemical shift as a reference point, and we have to align all the spectra along with the reference spectra also. So that thing, and only after that our data is ready for the multivariate analysis. Otherwise you can't do the multivariate analysis. The another steps, this is the same steps, let me explain it. you have to use, for that you have to use the internal control. And the internal control is that chemical shift, that concept I explained just in the last lectures. So to normalize or align the different spectra, you have, first of all, you know, from where you are starting the zero value of the ppm. Okay, here it is little bit different, starting zero, here it is a little bit different. So on the basis of that, you have to use the different, these type of the internal controls, and these internal controls can be start with the 01. TMS is the most standard reference material that we are using, and some computational approaches are also recommended to align the different spectra which are developed in the recent year. So now recently these positive control, we don't need them because we need some computational algorithm which can sort it out the usage of these internal references. So you can go ahead without using this thing now. So spectrum alignment was a computational issue, variation between the different spectra. It can be sorted out by using the reference materials, but yes, a few computational tools can give you the better recommendation for the same thing. So second issue that will come up with the spectrum pre-processing of the data analysis, that is labeled as a baseline correction actually. So baseline correction means that sometimes some metabolites are superimposed on the broad background that does not contribute any signal. So you know some shapes come this way. Some metabolites are present in that area, but you can't see them actually because they are overlapped by some errors, some other regions also. There is so many regions. So what is you have to do the baseline correction so that you can find out is there any peak or not. So any signal, but affect them. That's why we are using the polynomial fitting of the region in between the NMR signal that we are used for the automatic. This is an algorithm, but it is automatically, you just give a commands and they will do the baseline correction very well. So after baseline correction, now they cut the spectra actually. You think this is the spectra, but they truncated in this spectra and you only have to find out the signal of the metabolites only. There's nothing else. So whatever the peaks you are getting, after cutting after this particular baseline corrections, you will find out the area in which the different metabolites are present. Yes, these areas are starting from the 0 up to the 8 ppm. So 0 is a little bit not easy to say, so we are starting this scaling from the 0,1 to the 8 ppm. And this suppressor is also required to start with the acquisition. So sometimes water molecules, as we discussed in the suppression technology, so we have to use for this thing. So for water actually is giving the peak at 4.8 ppm. So we say we have an algorithm, we just click. So whatever the peaks that is present on the 4.8 after spectrum alignment, not before the spectrum alignment, after spectrum alignment, whatever the peak that is present at the 4.8 ppm, they reduce that peak at the 0. So now there is no peak of the water also. So this is another point that I would like to let us here. The third, that is the most important part of this NMR spectra, is the bucketing. You can use the another word also, that is the binning actually. So binning and the bucketing is very simple to understand. So this is just like a spectra, but if you do the bucketing, so you divide the spectra into small bins actually, small region actually, small scale. And you have to focus on this small scale actually. Better to focus, it would be better because if you focus on the whole spectrum, so it will be difficult for you to analyze all the metabolites. So we divide the whole spectrum into thousands of the small brackets, and you just focus on the variation along the different brackets. But it has one more disadvantage actually. Sometimes a peak can be split into the two different bracketing. So for that you have a few, but it does not give you so much falsified results. So we are going ahead with this one. You can change the size of the bucketing also, binning also, and this is basically the standard size for this one is the 0.04 ppm. So 0.4 ppm, 0.08 ppm. So whatever the peak coming here, we can analyze as an individual sector as compared to the whole spectrum. So what is the meaning that this process arrays the minor variation in the chemical shifting, and second most important that they reduce the data actually. So whatever the data that was the raw data, now it is the reduced, because we lost a little bit data here, but yes, we have to do the multivariate analysis. For that we have to use this one. Now, another important point is that the disadvantage I told you, sometimes the peak is split into the two different regions, so it is better to ignore those peaks, because if you try to cover those peaks, so you have to change the spectrum alignments and you have to start from the zero again. So sometimes we are using the adaptive intelligent concept beginning and the adaptive using for the wavelength transformation to do the automatic bucketing, and these bucketing are very good for us. Yes, now these bucketing can be carried out by the two software which are released by the two different companies in the NMR actually. Brooker is very well, they are using the MX, that can be a mix tool, and the ACDS NR manager. So these tools are coming along with the machine, so you don't need to do. But you need to understand that what is the concept and why this machine is doing what type of the things to give you the better data sets. So after bucketing actually, what happening is that raw data was present in the form of a peak. Now we have to convert the raw data that is present in the peak in some metrics actually. Because the whole data analysis is based upon some numerical values. So we convert that particular things into the different metrics, and just whatever this matrix is based upon the I multiplied by the J, data matrix S, and one row define the sample, another row define the chemical shift. So we have to make the matrix with respect to the defining the sample, one row, and second is about the chemical shift. So here sample 1 and the 2 and the 3, 4, 5, 6, they represent a particular row, while the chemical shifting is present in the different bucket 1, bucket 2, bucket 3, bucket 4. So this way, how many buckets are present, you have to use all the different chemical, you have to write down the chemical shifting of those different peaks actually. Suppose in a one bucket, there are, in one sample, in one bucket, there are 4.5, first bucket is 4.5, second is coming this way. So you have to do these experiments. Now many times what happening, you are doing the experiment in the replicates, okay. Two replicates are okay, but third replicates are giving overall higher value than first and the second one. So that you have to normalize it actually. So this intensify of the beam, the signal at the different chemical shift for the each sample. Disadvantage of this thing is that sometimes equally that the same thing that I told you, it is repeated again and again, split into 2 and 3, and that why you have to do it. Now adaptive intelligent bucketing, Gaussian bucketing, adaptive bucketing using the wavelength transformation and the dynamic adaptive bucketing. These are the four different concepts by which these metrics are developed. And they have some, every program is constructed by keeping one concept in my mind actually. If you are taking another concept, so another program is available. So we have to be clear what is our targets and what the things we want to harness to analyze, to find out, identify and analyze the sample here. So this is the normalization. Normalization is the fourth step of the NMR actually. What happening? Yes, two data sets are okay, but one data set is showing the higher value. So what should do? एक second. You can see here, everything is based upon some algorithm. These are the mathematical tools actually, metrics and everything. So here, forget about what the equation write down here, we will explain later on. So we have to focus on this matrix. So here you find out the sample 1, 2 and the 3. Here they are the bucketing 4.5, 2.6, 5.9, 6.8, 18.2 and the 5.3. But you find out the third sample has overall higher buckets. So when you use some algorithm, so what they are doing, they are making actually probability going normalization, and you will find out now the third one is the 0.22. After using this normalization equation, you able to make the things which is higher as compared to the other one. So now that is the normalized, and now your data is ready for the multivariate analysis. So normalization is a tool that is used in the LCMS and the NMR also. When I am explaining about the LCMS, so you will identify normalization is also using their one also. This is the equation, it is just giving the standard method to normalize the individual sample of the buckets, and this is just a different weight and the dilution result in the different concentration of the specific metabolites. So scaling is another concept actually. Scaling, what happening here in the class, you know very well, some students are very good and they are utilizing the 40 minutes of the teacher. Some student or the slow learner, they are not getting the attention of the teacher also, and they are not getting the enough time to interact with the teacher also. So in this concept actually, scaling, what you have to do, in the scaling, whatever the pattern analytes which are present at a high amount, so we have to reduce those amounts so that whatever the hidden metabolites, they can be come up and we can get the information about those metabolites. So scaling, whatever the present in the higher concentration, they contribute very strongly actually. So that's why we try to reduce them, and we are using an equation in which high concentration and the low concentration metabolites are converted into the some values which around the zero by subtracting with mean value from the column actually. So we have to assign some value, then subtract from the zero, and then we put another value. So these are the 4 or 5 different methods by which we can do the scaling. Yes, 4 different methods are using the 4 different equations. So we have to find out which one is the best with our experiment. So basically this pre-processing data was done by the company machines also. They have their automatic software. Those things I will discuss in very soon now. But yes, you have to understand this is a concept that is the necessary for our work. So now, this is the mean centric. So you have to calculate this one. These are the 4 different auto scaling. This is the matrix, and here 4 different auto scaling are giving the final where you can find out someone is the slow, low concentration, so they come up high, and some are present at the high concentration, they come down. So you can easily compare them actually. So that was the things here. You can see here. Here it is the 0.35, 0.2 and 0.22, but here you can find out this value change according to the different conditions actually, different algorithms. So this is the something that is another important point. So I am coming with the multivariate analysis. So I thought that Professor didn't cover this portion, but he took so much time for the LCMS actually. Yes, it's very difficult to cover the LCMS in one lecture, 2 and 3. It's required whole 2 and 3 days basically. There is no doubt about it. And it's a challenge for a teacher to cover one technique in the 1 hour in the 2 hours basically. So, but I also just come with a few slides of the multivariate analysis because it is associated with the NMR data also. So multivariate analysis, what is the meaning of that and why we need this thing actually? Whatever the data you are getting actually, so you have to use some mathematical tools. You have to use some algorithms, okay, to get some meaningful information from those data actually. So data is everywhere, height, come down, some up-regulated, some down-regulated. But if you are not using some chemometrics, so how can you get that really it is up-regulated or down-regulated? So for we have to do the multivariate analysis. And now this multivariate analysis is very common. You will find out in all the papers, whatever you are reading of the genomics, metabolomics, transcriptomics, and even the physiological data also. So this is something that is very useful for all of us. But I come here only a few slides for this thing. So basically we are using the 2 different types of the multivariates. First is the unsupervised and second is the supervised. So unsupervised is something that giving what is the meaning of the unsupervised? We are taking an example here, that is the PCA, Principal Component Analysis. That is an unsupervised recognition pattern methods, and what we have to do, whatever the sample we collected, we have to group them into the different, depends on our convenience. Okay, so sample based upon the signal that we are representing the metabolome. So in unsupervised, we don't have something in our minds that we have to discriminate them on the basis of height only. We have to discriminate them on the basis of the skin color. No. We have to find out what is the pattern, small, little bit, big. This way we have to differentiate them, separate them from each other in the different groups actually. So for that, this PCA is the best one actually. So here chemical shifting of the discriminating signal involved the distance of the sample can be identified by using the loading plots actually. So here the first thing you have to show that the your data has the different data sets which are different from each other. So means on the basis of some variation. So and second is that chemical shift of the discriminating signal can be easily calculated by the loading plot. So this PCA with the loading plots is giving the clear picture about the unsupervised pattern recognition in the data analysis. So this is a matrix actually. It is also in mathematics. So this transformation matrix that provide the information about the data after pre-processing named as a loading. So this is the loading and this is the principal component analysis. So this is the plot with respect to the two different variables with respect to each other. Suppose we have the data and we have so many variables. So one variable with the second with the third, one with the third. So this way we have to do series of the principal component analysis. Okay. Then we have to find out the pattern in between them. If we have the two variables, then it's okay. Otherwise, we have to repeat this experiment again and again. So in this picture, you can find out that there are only just two concentrated regions. One is the red and second is the blue. So these two concentrated regions is reflecting that both data sets are totally dissimilar with each other. So what is the meaning of that? You finally use some unsupervised recognition pattern to split the whole data into different sets depends on the similarity. So you are getting here. Now loading plots is doing that. Here you can find out how all bucket intensity are linearly combined to the principal components here. So here buckets 1, 2, 3, whatever we use, here principal component 1, principal component 2, principal component 3, principal component 4. So this way we have to make the loading pattern. In this loading pattern, you can find out that whatever the pattern, what is the meaning of that? Principal component analysis at the level of the buckets we are doing here. So you can find out one bucket is different from the another one. Earlier we were discussed about only the pattern at the spectral level. But now we focused on the small steps, that's the buckets, and in which we have to find out one variation, one principal component has how much pattern, second principal component has what type of the pattern. So you can get such type of the information by using the loading pattern. So one bucket, this principal component has the different pattern with respect to bucket 1, 2, 3 and 4, and it has the different pattern with respect to buckets 1, 2 and 3 and the 4. So that's why you are getting little bit more clear variations which can be differentiated from each other at the buckets level. So this one is the method. Another method that is the more important for all of us, that is the supervised method. In the supervised method, we have to go ahead with some already variation in our minds. So we know that we have to differentiate the sample on the basis of a few variables actually. So here supervised variable is basically help to identify some biomarkers. Biomarkers mean anything which can define that particular stage actually. So biomarkers are used for the cancer, so they can identify that particular disease. Biomarkers are used for the different disorders also. Similarly, if there is a 4 sample of the different stages, so you can identify what metabolites is present at what particular stage. So for that we have to do this thing. So this is a supervised one, and supervised one means you have to have the different clusters of the bucket representing a specific metabolite change only. Here we can find out how a single metabolite is changing from one bucket to the other bucket. So the wild type and the specific mutants is the example. This model is applied to the prior knowledge of the wild type and the specific mutants only, otherwise you can't do this one. Second, you have the prior knowledge of the sample classes. The third is that you can use to mark the separation between the different classes also. You must prior knowledge of the subject, analytes curve, then their class, and you know how one class is differentiated from the another one. And the another one is that separation between the different sample classes at the level of the individual metabolites also. 4 other different stages you have to take care for the supervised recognition pattern methods. Now it can be carried out by the 2 different methods. First is the PLSDA and second is the OPLSDA. PLSDA is partially square discriminant analysis and OPLS is an orthogonal partially least square discriminant analysis. So I will give you the, it is very easy to understand actually. So don't go ahead with the extra understanding. So just I was explaining this is the mutual regression method. First one was both are the mutual regression methods, and we have to use the pre-processed data, and newly directive y, x and the y are the algorithm, and mean value is the y for the wild type, and the for mutant we are putting the one value. So now we have to use this PLSDA, the x and the y plot. whatever the X and the Y plot we are using. So we have to use this algorithm actually. So this algorithm has the two components. This is the matrix of the sample Sx, and this is the X Sp plus my E. S plus my E actually, this thing can be replaced by this thing. This is the E. E is a residual part actually. It is present somewhere, but you will find out Xp in this formula. Basically, T represents the two things. First is the loading plots, which can P is the loading plot and T is the score matrix, which we cover in the first part supervised. This is the loading plot and this is the square matrix. So score matrix is coming and the P is used here. T and the P value we know. E is the constants, residual of every experiment. It is little bit different sometimes. So this is giving the information about the loading matrix as well as the score matrix. Now, if you go with the second one, so PLSDA is supervised method that differentiate between the group of the sample that are defined by the analytes. So now I want to differentiate all of you on the basis of how much money you have actually. So money is analytes and I can differentiate you. So when you know the variable, one variable, so you can differentiate that on the basis of the PLSDA by using this equation actually. Okay. Now, but most important information about the PLSDA, that is the correlation between the two data sets. One data set, second data set, how much they are correlated with each other. So that can be carried out for the group classification also. The OLSPADA has a little bit different from this one. Both are the regression. This is the PLSDA, this is the OPLSDA. Here what we have to do, the same matrix we have to calculate, but here we are getting this one also. XS0 plus E. E is the same residual matrix. And it can be put by, this X can be put by the TP, PPT plus T naught, P naught, T actually. So here we predict one thing actually. हमें पहले पता होता है कि some इनमें कुछ लोग ऐसे हैं, so we are putting them a different. We already know about those things. So that's why we are putting a filter. हम पहले से filter कर देते हैं कि भाई this is a, we have to filter out who are very height, who has very high height actually. So we are putting a filter in between this equation. When you are putting the filter, so whatever the person here, we can divide them into the two categories. One is already filtered out, second is the predictive actually, whatever you predictive. So OLSPDA has this filter and that's why it is also giving a different type of concept. So I am explaining here both the PLSDA and this OPLSDA components, so you will find out both are giving the different thing. There is no lot of the difference, but yes, because we are using the different equation actually. We are putting the filter here. If you know the filter, that 50% out of the 50, 40 will get the 100%. So 40. So you know their value very well. You just analyze the data on the rest of the data sets. So that's why this OLSPDA and PLSDA has the depart from each other, but they have the different patterns. But if you go with the loading pattern, loading plots actually, these ones. So you are getting the little bit different from it. Here, principal component analysis 1 and the 2 and the different buckets. So you identify that PLSDA is giving the information in a different way, but when you are going with this OPLSDA, so you are getting the different pattern actually. So that sometime, and even whatever the classification, this data analysis, multivariate data analysis is just to analyze the data, classify data based upon variable. Whatever you are using classification method, all come in multivariate analysis, and these are the algorithms which we use to classify them, to demarcate them from the another one. But the most one that for the unsupervised we are using these two methods, that is the PLSDA and the OPLSDA. So this is all about the NMR, and yes, now I am coming on the second lecture of, that is the most